Good evening, everyone, and welcome. Thank you for logging in early. While others are logging in, I will go ahead and start our introductions for today. My name is Farah Amro. I am a graduate admissions counselor at the Halmos College of Arts and Sciences at NSU. Thank you all for joining us for another session of the Halmos College Virtual Lecture Series, where we highlight research conducted by our own students and faculty across the sciences. Today's lecture will explore how rising temperatures influence interactions between aquatic hosts and the parasites that live within them, an increasingly important topic as warming events continue to affect marine and freshwater ecosystems. Before we begin, I'd like to briefly go over the flow of today's session so everyone knows what to expect. The lecture will run from 5:30 to 6:30 p.m. The first 40 to 45 minutes will be Briana's research presentation, and then the remaining time will be dedicated to your questions. To keep the discussion organized, we ask you to please submit your questions through the chat. And at the end of the presentation, those questions that are related to the research itself and the lecture will be answered by our presenter, Mrs. Briana Zaffiro, and questions related to the department itself, the Biological Sciences Department, or the admissions process to any of our biological sciences programs will be answered by our Assistant Director of Graduate Admissions, Mrs. Megan Choi. Please feel free to submit questions in the chat at any time during the lecture, and we will review them during the Q&A portion. And as I mentioned before, Mrs. Megan Choi is joining us today. Megan works closely with applicants to our biological sciences graduate programs, and she will be available during the Q&A portion. She is the best and the kindest person to guide you through admissions, so she's your person. Thank you so much for joining us this evening, Megan. And it is now my pleasure to also introduce today's speaker, Mrs. Briana Zaffiro. Briana is a Master of Science candidate in the Biological Sciences at NSU. Her research focuses on how elevated temperatures influence the relationship between parasites and their hosts in aquatic ecosystems, particularly how thermal stress can affect both parasite activity and host resilience. She earned her Bachelor of Science in Marine Biology from NSU and plans to continue her research through a Ph.D. program, with the long-term goal of leading a research laboratory of her own. Today, she will be presenting her work titled The Effects of Thermal Stress from a Host and Parasite Perspective. Briana, thank you for being with us today. We're looking forward to learning more about your research, so I will now turn the presentation over to you.
Thank you, Farah. So as Farah said, yes, hi, I'm Briana. I'm a Master's of Biological Sciences student, and I'm talking about the effects of thermal stress from both a host and a parasite perspective. And so to start this off, I'm actually going to go a little bit through this presentation outline. So this is going to start with a general introduction, and then talk a little bit about the fish perspective of elevated temperatures, the parasite perspective, a general conclusion, and then I'm going to talk about some potential next steps, and then I'll have time for acknowledgements as well as questions. And this is really split up in two major parts. The first one's going to be the fish perspective, and the second part's going to be the parasite perspective. And both of these are going to be marked with either a blue fish or a pink worm. And when you see both of these on the screen, that means that I'm talking about both host and parasite. And so with that, we're going to go ahead and get started. And so temperatures have been steadily elevating over the years. And with those also comes these extreme thermal events that have been occurring in a much higher frequency. And one such example of this is the summer event that happened in 2023. In 2023 in South Florida specifically, we saw these really high spikes of temperature in our ocean, of ocean temperatures, especially in coral reefs. And this really drove a lot of this coral reef bleaching. Bleaching is when corals will expel their dinoflagellates, which is their main source of food. And this usually ends up happening is these corals will not be able to feed themselves and will eventually die off. And in this specific 2023 summer event, there were some areas in South Florida that actually experienced mortality rates upwards of 100%. And this is just one such example of a disease that is affected by elevated temperatures. Another example is this little copepod called Argulis foliosus. And so this is a copepod that has a seasonality-based life cycle, where in summer months they'll be much more active, and in winter months they'll be much less active. Um, and as summers get longer and winters get shorter, they actually will have a very high level or much higher level of activity than they usually do. And they might look a little small, but they might look small and even a little cute. But in as of 2006, they cost salmonid fisheries about $305 million a year. And granted, this is also a number from 2006, which was 20 years ago. So this has only increased as temperatures have increased. Another example of a disease that is affected by elevated temperatures is these amoebas that are called Paramoeba invadens. So this is an amoeba that's found in Nova Scotia and is usually infecting these green sea urchins on the left here. These green sea urchins are a pretty important part of the ecosystem in Nova Scotia. And these Paramoeba invadens, so they have a critical thermal minimum of about 2.5 degrees Celsius. What this means is if the temperature goes below 2.5 degrees Celsius, they won't be able to survive it and they'll actually start dying off. Nova Scotia has not reached below 2.5 degrees in quite a while, so these Paramoeba invadens are not experiencing seasonal die-offs, and these green sea urchins are really their populations are being really negatively affected by the increase of these amoebas. And if this pattern continues, it's predicted that these amoeba might cause the functional extinction of these green sea urchins. And so I've been talking a lot about parasites, so I kind of want to go through a little bit and explain what exactly is a parasite. So according to the CDC, a parasite is an organism that lives on or in a host organism and gets its food from or at the expense of its host. And usually when we think about parasites, we'll either think about the really long tapeworms that you can see out of bear butts, or the 6-foot tapeworm that comes out of a guy who really likes to eat fish. It's more often than not, a lot of parasites, their hosts can't even feel that they are taking energy from them or taking food from them. And so, for example, this is something called a Demodex mite. And so this is a little small mite that is actually lives on human eyelashes. So if you're watching this, that means you have about a few million of these on your eyelashes, and you most likely wouldn't have known if I didn't tell you. So, surprise. And so other than that, they typically have a multi-host life cycle, and this can be used as environmental health indicators, and this usually with, yeah, sorry, this can be used as environmental health indicators with high community diversity being associated with healthy environments. And what this means is the more different species of parasites you have, the more healthy your ecosystem is. And so an example of this multi-host life cycle is going to be in these flukes or these digynians, which is a type of flatworm. And so it starts as an egg that's excreted from a bird. Once this egg hits the water, it'll grow into this mercidium free-living form. This free-living form is going to start swimming around and trying to find a snail host. And once it finds its snail host, it's going to infect the snail and then grow to a sporocyst, which is a parasitic parasitic stage that stays within the snail that houses these cercaria. And so the cercaria will grow within the sporocyst, and as the sporocysts clone themselves and multiply, the cercaria will start to leave the sporocyst and start infecting the snail. Once they are mature enough, they will actually leave the snail and get into and become a free-living version of cercaria, where they have a tail and they start swimming around until they find a fish host. Once they find a fish host, they'll penetrate between the scales of the fish, encyst within the fish, and stay there until the definitive host or the bird comes and eats the fish. Once the parasite is eaten along with the fish, it'll leave its cyst and grow into its adult form within the bird, and it'll start laying eggs, and the cycle will continue again. And so through this one species of parasites, there's quite a few different hosts that it is in contact with, and we can use that information to look at different links between different hosts. And so it's a one, yeah, we can use it to look at different food webs, which is how different animals eat each other and how energy moves throughout an ecosystem. And there was a study done in 2005 where an aquatic food web was considered without parasites. And so they pretty much looked to see which animals were eating which and how did they connect to each other. This is without parasites. But when you add in parasites, you can see there's quite a few more connections made. And so I'll go back so you can kind of see it as well. This is without parasites, and this is with considering parasites. So there's quite a bit of information that is being missed when food web studies don't consider parasites in it as well. And with that, we are going to go and start into the fish perspective. So um, the host that I used for this study is the common chiner, and the common chiner is a popular bait fish, and these guys are native to northern parts of the U.S. and Canada, but they're also reared exotically in places like Florida and Georgia. They typically eat small insects and smaller fish, but as they usually are reared as bait fish in Florida, their main diet will consist of commercial flakes commonly used in aquaculture. And so just like any other fish, these guys will experience thermal stress when temperatures rise. And when elevated temperatures do occur, fishes may experience something called loss of equilibrium or LOE. What happens here is a fish's muscles will begin to spasm. And when they begin to spasm, the fish can't control them properly. And so they're not able to righten themselves. And so this looks like the video on the right where this fish can't really stay upright. It's kind of spinning a little bit. And over time, as these fishes are getting more and more used to high temperatures, they'll actually end up shrinking in size as they'll be unable to withstand in a heightened energy demand. And so this idea, we're there's been quite a few studies on how high temperatures affect fish, but there hasn't really been a lot of information on how this looks with naturally occurring ecological diseases, such as these small parasites that may influence this reaction to thermal stress. And this is kind of the idea of if something has more parasites, is it going to be able to withstand heat less, or is it going to be better at withstanding heat? And so to assess this, I ran some trials called CT Max trials. And what this consists of is putting a fish at a consistent acclimation temperature and then slowly increasing the temperature until the specimen hits that loss of equilibrium or that LOE and they're unable to righten themselves. At that point we consider that our endpoint of okay, at this temperature the fish is unable to maintain its normal behaviors and is unable to swim. And so to do this, I set up a bath where it was connected by a net in the middle. On the left side, I had the fish in there, as well as a few sensors. On the right side, I had a bubbler as well as a heater. The heater made sure that the heat did increase over a consistent rate, while the bubbler made sure that the bath was highly oxygenated and it wasn't oxygen that was causing the fish to experience LOE. And then once they would go through these trials, these fish would be reacclimated to that 20 degrees Celsius, set aside for 24 hours so we can see if there's any trial-related mortality. And this is the LOE again, and then the fish were then dissected for parasites, and parasite intensities were determined for each individual. And so when we did that, this is what we found. So we found a few different types of parasites. We found one called Schizocotyledon aglenafi, which is a tapeworm that's seen in the top right, as well as a digynian called postdiplostomum, which is seen in the bottom right. We also found some Capillaria species, as well as a larval acanthocephalin. And so with this information, we were also able to see how the fish's the CT max or how the fish's CT max related to their parasite load. And so what we really saw was the more parasites a fish has, the lower its CT max. And so for example, here we had one fish that was infected by 120 parasites that only was able to withstand up to 35 degrees Celsius, while a lot of our fish that weren't infected were able to withstand much higher temperatures, upwards of 38 degrees Celsius. And other than that, we also have a few a few parasites of note. So the first one is a Schizocotyledon aglenathi, that's also called Asian fish tapeworm, or AFT. And so this is an invasive tapeworm that's spread throughout bait fishes. And in common shiners, AFT is found as an adult within its viscera. AFTs also has a very large range that spans worldwide and is very highly pathogenetic in a lot of the areas where they're found exotically or outside of their native range. AFT may infect fish that have intestines the same size as the worm itself. And so we saw this in these common shiners here, where a lot of these tapeworms were literally the same size as the fish intestines, if not bigger. This does lead to a lot of mortality, as when these tapeworms get big for the fish host intestines, they'll actually burst the intestines and cause the fish to die. So this definitely increased or decreased the fish's ability to withstand temperature. Other than that, our most commonly found parasite was this Postdiplostomum species. So this is a genus that is commonly found in bait fishes, and it's also one of the digynian taxa that cause black spot disease. In common shiners, it's found throughout the viscera and on top of the brain, which leaves potential for parasite-mediated behavior. And so this parasite-mediated behavior happens when a parasite infects a fish and pretty much influences it to do a behavior or go into a certain spot that will allow it to get eaten by its next host easier. So, for example, what this might look like, if there's a small fish that's infected with this Postdiplostomum, and the Postdiplostomum, to reach its adult stage, has to be in a bird, it'll actually influence the fish to swim towards the top of the water, so a bird is able to eat it easier. And yeah, and so the mechanism behind this, though, how these parasites influence their fish to do different behaviors, we don't quite know what causes that yet. And so, other than that, we can pretty confidently assume that temperatures above 35 degrees Celsius will result in common shiners in South Florida with high intensity infections to not be able to withstand temperature spikes when they do occur. And however, so a lot of these common shiners that have these 100 plus infections of Postdiplostomum especially will not be able to withstand those lower spikes. At temperature spikes higher than 35, or higher than 38 degrees Celsius, this may result in entire populations of common shiners disappearing. So however, we can assume this, but the physiological mechanism behind this association of parasites to temperature, we don't quite know yet. And so with that said, we are going to go on to the parasite perspective. And so for the parasite perspective, we're first going to talk about the environment of a parasite. So these parasites experience an environment pretty differently than we do. We have a pretty high level of oxygen when we're just walking around, but these parasites don't get that same level of oxygen. So, for example, a tapeworm in an intestinal tract will experience a different level of oxygen based on where within the intestines they are. So, for example, if they're in the small intestines, they're going to experience more oxygen, but if they're in the and yeah, and if they're in the end of the small intestines, they're going to experience less partial pressure of oxygen. Where if they're closer to the capillary vessels or they're closer to the wall of the intestines, they're going to be experiencing more oxygen than if they were in the middle of the intestines. And what this results in is these guys being called, or something called facultative anaerobes, which means that they're able to switch between anaerobic and aerobic metabolism. And so anaerobic means without oxygen, while aerobic means with oxygen. So depending on oxygen availability, they're able to do that switch back and forth, or they're able um, to still be able to function with or without oxygen. And so with that, there also has been some work done on parasites and elevated temperatures. So these Cercaria, they are a non-feeding life stage, and how they usually get around and how they're able to swim into their next fish is they will have these glycogen stores that are within their tail. And as temperature increases, their activity level will increase. However, as this activity level increases, so does mortality. And this is thought to be because they are eating their glycogen stores much quicker. And once they eat that quicker, or once they use that quicker, they're unable to continue to swim around. And so here you can see this is in a study from Morley 2011, where the higher the temperature goes, the more glycogen is utilized by the Cercaria individuals. And so to assess this idea of temperature, yet to assess this idea of uh, temperature-based metabolism, we first collected these dark Sarah snails, and they were collected off the coast of the intercoastal, and then brought back to the lab, crushed and dissected for parasites. And I was looking specifically for this parasite called the bivesiculid. So this family bivesiculid is a group of digynians with Cercaria that are actually consumed by their fish or by their definitive host. Most Cercaria will penetrate through fish scales, where these guys will actually puff up their tail around their main Cercarial body. So they look like they're a dying fish. And so their definitive fish host is able to see what they think is a struggling fish, and they think, oh, this is easy prey because this fish isn't going anywhere. But in actuality, it is actually a parasite. And so these parasites have a slightly different life cycle than the Cercaria or the digynians that I highlighted before. So they start as an egg within their definitive fish host. This egg, once excreted and once they go into water, will turn into their next free living form, which is the marsidium. Once this marsidium goes and finds a snail, it'll then grow into its sporocyst stage, which then grows into this radia stage. This radia is almost like a sporocyst. It is the same protective type of organism, but this stage has has organs and can eat, unlike the sporocyst. This radia will contain the Cercaria within it that will then leave the radia and infect the snail. And then once this Cercaria leaves the snail, it'll go into its free living form where its tail will puff up around it to be eaten by the fish, which will then reach its adult form within its definitive fish. So unlike the previous digynians, these guys only go between a fish and a snail host rather than having a bird host as well. And so to do so with these Cercaria, we set up a water bath that's able to keep a consistent temperature throughout each of these runs, which was attached to a amp with a O2 logger right next to it. And so within the water bath, we kept these chambers. And so these chambers housed the specimen. So within a glass syringe, we had a media that's previously been shown to keep these bivesiculate cercaria alive for more than 12 hours, as well as about 20 cercaria specimen. And in this syringe is also a Clark's polygraphic microelectrode. And so this is covered by a semi-permeable membrane with an electrolyte within it, and how we're able to measure the specimen's oxygen consumption is the oxygen that is within the media will flow through the semi-permeable membrane and react with the electrolyte that is touching the electrode. Um, the the platinum cathode will then pick up um, the energy that this lets off, and it'll be read as a microvolt. And so that looks like this output. So over time, we'll have this long output of different recordings of microvolts. And how we analyze this data is we take this first section as our high calibration, this next section as our low calibration. This for the first 30 minutes, we let the parasites kind of acclimate to their chamber, into the new environment. And then this is our actual data that we use to analyze. And from here, once we were able to take the oxygen consumption from these worms, I took our data and I fitted it to something called the Sharp Schoolfield model, which is used to describe the thermal performance curve of these bivesiculate cercaria. And so what resulted was um, was this was this amount of oxygen consumption throughout different temperatures. So at 20 degrees Celsius, we had the lowest amount of oxygen consumption, and at 35 degrees Celsius, we had the highest amount of oxygen consumption. And what you can see here as well is not only does oxygen consumption vary, but also there's a increasing amount of variation of each trial as the temperature goes up. And so this may be caused by individual differences, by individual differences as well as population level differences. And so these cercaria, as I mentioned before, depend on stored glycogen within their tails. But this level of glycogen will vary by individual, with healthier individuals having a little bit more glycogen than unhealthier individuals. And yeah, and then population level differences can be may be explained by the amount of crowding within each snail, as each snail will have infections from anywhere from 300 to 1,000 individual cercaria. So depending on that number, these cercaria may be healthier or not as healthy. And once they leave their snail, they may be able to breathe different amounts of oxygen and go through their glycogen stores at different rates because of that. And so other than that, this is the thermal performance curve that we were able to get from the oxygen consumption data. And so as you can see, it follows the same pattern of rising up to 35, maybe even a little past 35, and then falling right after 35 to that 40, which shows that our peak activity level of the cercaria is right around that 35 degrees Celsius. And so with this, we're also able to determine the caloric demand that each bivesiculate cercaria needs to use each day. And so we were able to get this using the oxycalorific coefficient for glucose, and this is expressed as kilojoules per day. And so these kilojoules is the amount of heat that these cercaria give off per day. And so each individual will have a differing amount of calories by temperature. So at lowest temperature of 20 degrees, each individual needs about 0.001 kilojoules per day. And then at the highest temperature, they need about 0.013 kilojoules a day. And these are very small numbers for each individual, but it still has a 900% increase from that 20 degrees to that 35 degrees Celsius. And so one thing about Cercaria in snails is you'll never find an infection that's only one Cercaria individual. Usually there'll be hundreds of individuals upwards to 1,000. So if we were to look at a lower infection rate or a lower infection in a snail, these caloric demands would increase up to, for 20 degrees, 0.43 kilojoules per day, and in 35 degrees, it'll increase to 3.89 kilojoules per day. And now, let's say we have a really high infection of these parasites in a snail, and there's 1,000 individuals in this population. That caloric demand in 35 degrees Celsius rises up to 12.97 kilojoules per day. This is a huge amount. And so snails typically need about 20 kilojoules of energy a day that they need to use or that they need to burn off. So this is more than half of what those snails already need to use, which is also increased in higher temperatures for snails. So this is a demand that these snails most likely will not be able to withstand as time goes on and as temperatures rise. And so, other than that, kind of looking at both of these parts together, we can we can we can say, or yeah, we can say that as temperatures increase up to 35 degrees, these or degrees Celsius, these parasites will benefit while their host will not, which will leave their host at a disadvantage. And in these really extreme temperatures above 35, both parasite and host will be unable to withstand temperatures, which will leave both host and parasite at a disadvantage. And so, other than that, some potential next steps that we would be able um, to do after this study is looking further into the mechanisms of what describes a fish ability to withstand temperature with a lower, looking further into the mechanisms of what decreases a fish's ability to withstand temperature with a higher parasite load, kind of looking at what's that actual link between between increased thermal stress and increased parasite load. And then other than that, uh, another step that we could take is it's investigating the smaller cercaria that typically penetrate host in this fettings media that we used in the second half, as well as comparing their oxygen consumption across temperature regimes to cercaria of bivesiculate species. So kind of looking at the normal smaller cercaria that instead of puffing their tail around them and swimming, are able only penetrate hosts and have a little bit less time in the water than these bivesiculate species do. And other than that, I have a lot of people that I need to thank for helping me with this project. My committee, especially with Dr. Blunar, Dr. Martinez, and Dr. Kerstetter, as well as Dr. Kate Sheehan, who gave me the recipe for the fettings media that I used, and as well as my lab mate Will, who allowed me to share a host system with him. I also have a lot of my friends that I need to thank, especially Mary, Paige, Jillian, Jacob, Heather, Aya, Perry, and Sarah. And I also need to thank some undergrads that helped me with this work. So Charlotte, Natalia, Michelle, thank you. Also my parents for their for their financial and emotional support in this degree. And I lastly need to thank my coworkers and friends at Fairchild for allowing me to talk way too much about worms. And with that, I am able to take any questions.
Thank you so much, Briana. That was excellent, fascinating, and pretty deep for tonight. I actually, when we hear about global warming, there are a lot of outcomes that we hear about, but this one I've never heard of personally before. And it was really interesting to hear you talk about that and how The temperature shifts can influence host physiology, parasite dynamics, and in turn can affect our aquatic ecosystems. I have a question for you while people are, putting in their questions. When you were doing your background research, I'm curious to know if you found any findings in human beings that were similar to what you found in fish or what you were trying to find in fish. Like, have you ever read anything in human beings where parasites also change the way that the relationship between the parasite and the host, which is the human being in that sense, is affected by the rising temperatures? Did you find anything like this?
Yeah, so I actually I mainly focused on fish, but I did come across some human-centered studies.
Mm-hmm.
It was mainly in parasites like what are they called? Schizosomes?
Okay.
Schizosoma? Yeah, those parasites. But
Okay.
those are also a fluke that experiences or they will um change similarly to or their behavior will change similarly to these bifasciculates where in increased temperature they'll actually be more active. And so there's a higher chance of more people being infected as temperatures rise,
Mm-hmm.
because the more a parasite's able to swim around and look for their potential host, the higher chance that they're going to find that host.
Right,
So
right.
I've definitely seen a lot of that.
Okay.
And the same for some other flatworms as well, but that was the biggest one that I saw.
Mm-hmm. That's interesting. Here's a question. Your research looks at the system from both the host and the parasite perspectives. Why is it important to study both sides of this interaction?
Yeah, so I find as the more I study parasites, the more they're taken for, or I guess not really taken for granted, but they're not the more they're not really looked
Explore
into
that.
as much as they should.
Yeah.
Um, Parasites are in everything, right? Theoretically, everything and everyone has a parasite, like us with the mites.
Mm-hmm.
And a lot of these environmental effects and a lot of these natural effects, um, they just looking at them from the host perspective doesn't tell us a ton about what's actually going to happen in the fields or in nature. So these fish, like the fish in my first part, I didn't go in and I didn't infect any of them. They were all naturally infected like that. Uh,
Okay.
so it's really important to consider these their parasite communities because they do have a influence on them even if we can't physically see it. Um, but yeah.
Okay. Another question, Brianna. Your research shows that higher parasite loads may reduce the thermal tolerance of fish. What ecological consequences could this have for fish populations as aquatic temperatures continue to rise? Where do you see this
Yeah.
going?
Yeah, so for a lot of these, I mean, a lot of these fish that are unhealthy or not unhealthy, but have higher loads of parasites, those populations are going to shrink. And those, as well as that parasite diversity, is the most important thing, is going to shrink as well. And these parasites that are in each of those fish, like especially the common shiners, they do fulfill a certain niche within that common shiner that that parasite has evolved to work or not to infect that fish and to grow with that fish. With, for example, the postdiplostomoms,
Mhm.
if all of those high intensity postdiplostomal infection fish die off, that might leave room for another more harmful parasite, for example, like the Asian fish tapeworm, where they more often than not result in fish mortality. So in um, in the present, as those not in the present, but in normal temperatures, we usually have that pretty wide diversity of parasites that with those specific niches, the host um isn't being harmed as much as it could be. But as those high intensities and as those large numbers of parasites die off, then that leaves room for some more harmful parasites. And
Right.
with the common shiners, they're a small fish that are also eaten by a lot of other bigger fish down here, right? So that not only affects the shiner itself, but also what eats the shiner and what eats that fish and what eats that fish. So it has quite
All
a
that
few.
food web that you described.
Yeah,
Whole
yeah.
right. And I believe there's a question. So do you see any potential applications of your research for fisheries management or aquacultural systems? And I believe, the answer is yes, but go ahead and tell us how.
Yeah, absolutely. So, and this is I focus on Cercaria here, but there's a lot more fisheries specific parasites that this can be applied to. For example, that Argulis foliaceus that I looked at or that I spoke at about before, looking at how that that copepod's um, thermal performance changes with as fishes change. Again, that costs us it costs fisheries quite a bit of money that 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 number was in both fish loss and um trying to prevent um and not cure but trying to prevent that disease from happening. So it absolutely stands for a huge loss fisheries wise if this idea isn't really taken into account for.
Uh, Last question for you, Brianna, and then we move on to questions for Megan. As a graduate researcher working in this field, what has been the most surprising or challenging aspect of studying host-parasite interactions?
Ooh, that's such a good question.
Mm.
That is such a good question. I honestly, the a lot of this research and a lot of this study was very novel in its approaches and its methods, and just kind of figuring out what might work and and um and so for example with these parasites no one's really looked at metabolism before, and what metabolism is looked at is very theoretical and very kind of broad and vague. So no one's really been able to, or no one has looked at specifically oxygen consumption, especially with these parasites that switch between anaerobic and aerobic metabolism. It's hard to find a way to look at its full metabolism. And with this worm or with this parasite, I did get a little bit lucky because these are quite bigger than most Cercaria are. So this is about like 5 times bigger than some like than some of the smaller Cicaria that penetrate fish hosts. So just kind of having to brainstorm my way through that was definitely fun.
Yeah,
It's
more research
definitely a challenge.
because you spoke
Yeah.
about certain interactions, but we don't know how they're happening even. So there is more research for the future. Is like how is it affecting the fish to change its, when you talked about fish moving toward the another host so that the parasite can infect another host. That was also interesting. So that so
Yeah.
there's
Yeah,
definitely
and definitely keeping animals alive is always a fun
yeah.
hurdle to get through.
So Brianna, before I ask Megan, one last question to you as a graduate student in the
Yeah.
Department of Biological Sciences. Tell us a little bit about your experience, how the faculty, was it easy to do your research really, to get resources? Tell us your experience as a graduate student at NSU.
Yeah, yeah. So I definitely wish I could say that research was easy. It is not. It's, I as a marine not marine, sorry, as a biological sciences student, I actually had a lot more time to focus on this research. So something that a lot of my marine science peers don't or experience a little differently than I is I have less classes than they do. So I really am able to spend so much of that time going through all the literature and making sure I know what methods to use and making sure that I can collect all of my data. And then I have also been very fortunate to have financial support from from the college as well. So a lot of my ingredients for my cell culture, for example, was all funded by the Department of Biological Sciences, and I really couldn't have really even kept these worms alive overnight without that. Um, but yeah, it's definitely been definitely been very helpful, especially with a lot of these. My thesis itself had a few changes in it, as most research projects do go through. So I was able to pivot quite quickly and quite efficiently with the support that I got from the biological sciences department.
Awesome. Well, we're happy to hear that. Brianna, I'm going to move on to Megan. Megan, are you ready for us?
Hello, hello. Brianna, that was wonderful. Thank you so much for sharing your research with us. I know a lot of the times we're asking you guys before you're ready to graduate and present to your committees. So we're always excited to hear the work that you guys are doing. So I want to thank you again for being a part of this, our virtual lecture series for our winter term, of course, our student series. But I also want to invite any of our guests that are here, if they have any questions about the program that Brianna is a part of. I love the fact that you mentioned about having funding for some of your research materials. That is a big portion of things that students are looking for when they're looking for master's and PhD programs is what type of funding is available to them. So I'm happy to hear that you were able to utilize the funding for some of your research materials. That's wonderful. I do, like I said, I want to open it up to our guests. If anyone has any questions about the master's program that Brianna is a part of, or if you have any questions about just the admissions process in general, we're happy to answer any of those questions. We'll give you a minute or two.
Should I keep sharing my screen or should I?
And that's fine.
I guess it is the question slide, so it kind of works here.
Good.
Our little friend Parasite.
Yeah.
Okay, so Megan, a question to you. I'm interested in applying to the Biological Sciences graduate program, and research is really important to me. How early do students typically get involved in research at NSU, and what kind of opportunities are available?
Wonderful question. So, what I like to tell most applicants is that the wonderful thing about our master's degree program is that you don't have to come in to the program with a solidified idea of what it is that you're going to do your research on. You can have a general idea, a general subject area, an area that just interests you. And we give our students, it is a 2-year academic program. So all of our students that are in the research department can finish their academics within the 2 years. And then we give you a little time to finish your research if it takes a little bit longer. But we give you that first academic year to determine your topic of research. So throughout the program, you're going to be producing bits and pieces of your research. When you get to the point where Brianna is at, where you have all of your materials, your research is kind of done, you'll build your presentation and finish out your thesis. So for students that are interested in our program, you have many options. One of the things that I recommend is that you look at the faculty members and see what areas of research they are in. If you find a faculty member that's studying something that you have interest in, like let's say genetics, you have an interest in that, and you find one of our faculty members who's working in genetics, you can reach out to that faculty member even through the application process to talk to them to see if they would be interested in working with you if you came on board into our research program. Some of our students that are a part of the research program even have the opportunity to work in labs. Whether it's a lab focused on the area of research that you're working on, you still have an opportunity to work with some of our faculty members if positions in their labs are available. So there are lots of different opportunities for research, whether it's your specific research you're working on for your thesis. Every student has a faculty advisor that they will work with to make sure that their research is moving throughout the program. Or you have opportunities to work in some of the labs that we have on campus as well.
Okay, another question, Megan. If I were to join the program, would there be opportunities to collaborate with researchers or students from other scientific disciplines within the college? And I do believe that Briana already mentioned that she did work with undergrads.
Yep, and
Yep.
as for Brianna's research, she is in the marine world. So while she is a student in the biological sciences, we do have some crossing over with our Department of Marine and Environmental Sciences. We also have some crossover with our Department of Chemistry, Engineering, and Physics. So depending on, again, what it is that you're interested in working on, there's opportunities to work within our department and also crossing over with some of the other departments within Halmos College. And then some of our students also have the opportunity to work with our undergrads. So we have some teaching assistantship positions that are available to our students that are in the biological sciences. And in that, you're kind of building your own resume while working with our undergraduate population that are in our biology department.
Megan, for someone planning to apply, what are the main parts of the application that the admissions committee focuses on the most?
Okay, wonderful question. So I think a lot of the times applying to graduate school can seem a little scary. What I love is that the biological sciences department in the last couple of years has tailored down what they are looking for in students to make it a little simpler to apply, but also to make it so that our program is not unobtainable for students. So for the application itself, we are only looking for your official transcripts, your official transcripts and your resume. For our students applying to research in particular, I ask that on their resume, they indicate like in an objective statement, some type of research that they're interested in. And if it's one or two or three areas, name them all. That way, when our admissions committee is looking over your application, they can kind of have an idea like, okay, this student has an interest in parasites, in microbiology. The student has an interest in genetics or genomics. They have that idea going in as they're reviewing your application. We are looking over your science coursework. Of course, you need to have your general biologies and general chemistries already down. Those are the foundation of what you will build your academics on. But we're also looking for some higher level coursework. So if you are that student that's like interested in genetics, we want to see that you've taken some an upper level undergraduate class that can give you the foundation that you need to focus your research in genetics. If you are interested in research that maybe will take you off to a PhD, let's see that you took a couple of your upper level coursework in maybe statistics, because we have some research that you have to do. Statistics is a class that you might need in order to complete that. So it does depend. It can kind of be tailored student by student. And the wonderful thing about our department is if a student has joined our program a little later or they are building up their coursework, we will review all of your science coursework. We will see how you did freshman year versus how you've been doing junior and senior year. And our faculty is willing to work with our students. So that's the wonderful part of this program.
Okay. Megan, for those admitted, how do those admitted apply to teaching assistant programs and what do those positions usually involve?
Wonderful question. So for our application process, the research program in and of itself admits students for fall, summer, and winter term. For the hiring of our teaching assistants, we hire in the April timeframe. So generally admitted students are information is sent out by our graduate academic coordinator, Gwen Hauer. She will let students know when the application is open. We open it every March, and then students can apply at that time. You have to be an admitted student, so that means have your application already submitted in order to be considered and interviewed for this position. The wonderful thing about the position is that our teaching assistants work fall and they work winter term. So 2 terms every academic year. And the TA position comes with a $10,000 award, $5,000 of which goes towards your tuition, and the other $5,000 is provided to you as a stipend paid during the semester as you are as you TA. So for a student that is in our academic program, in one academic year, you can earn up to $20,000 as a TA.
Amazing. Okay. Megan, as an incoming student, what's the best way for students to build relationships with professors in the program?
Wonderful question. I always say reaching out to them. I have never met a group of faculty members that love to talk about research and what they are doing than this bunch. So if you are a student that has an interest in something that one of the faculty members is working on, reach out. Introduce yourself. Hi, my name is Megan. I'm going to be an incoming research student this upcoming fall semester. I'm really excited about the classes that are being offered. And I saw that you are teaching advanced anatomy. And I wanted to talk to you a little bit more about the project that I'm interested in and to see if maybe you could work with me through my time in the program. Simple email. We also have through Gwen Hauer, once you are an admitted student and you've solidified your seat in the program, then we give our students an opportunity to meet with Gwen to go over their coursework. Gwen is also a faculty member. So the time in which you talk with her, she can give you a little insight about which faculty members are great to reach out to.
The website mentions both a thesis track and a capstone track. How do students decide which option is the best fit for them?
So when students are admitted into the research program, you all will start as a capstone student. Through your first academic year, that will be your determination of whether you're going to do a capstone project or if you're going to complete a thesis. Students don't have to make that decision during the first semester, but by the end of their second term, that is when they will make the decision on which track they're going to stay on. The only difference in coursework is the research or the capstone course. So all of the coursework is still the same for both sets of students, but then once you've made the determination of which is better for you, and again, you'll work with your faculty advisor, you will work with Gwen, and you'll work with the program director to make that determination for yourself of which track might work best for you and the information and research you're wanting to do.
Thank you, Megan. Debbie is asking, how does the alumni scholarship apply to NSU alumni?
So that is done outside of the admissions department. All scholarships are done in one of three levels, either through the academic department, so the Department of Biological Sciences. When they have scholarships available, they will let all admitted students and current students know so that they can then apply for them. The college itself, Halmos College of Arts and Sciences, they have a scholarship website that also includes institutional scholarships as well. So all scholarships are done outside of admissions, but are listed on our university website.
Okay, this was our last question, which is, for students who complete the MS in Biological Sciences, what kinds of career paths or opportunities do they typically pursue afterward?
That all depends on the student. So we have a wide variety of areas that our students have gone into. Some of our students will go off to PhD work. So they will either end up in academia because they will continue to teach while they're doing their PhD, and then after. Some of our students will go into research. They will become research assistants, research scientists. We have some students that will go into PhD work in other areas as well. So not just in biological sciences. We had a student that recently graduated going into a PhD program in at a college of pharmacy at another institution. We have a student that currently works for the National Institute of Health. So there are different areas. Bioinformatics is an area that is starting to grow. We actually carved out an entire concentration just for bioinformatics based off of some of the jobs that our past graduates have gone into in that area. So there are quite a number of different areas, just depending on what it is that your interests are. But academia is one, general research is another, and then of course teaching. We've got so many of our students that enjoy being TAs that they've stayed on with us or at other institutions to become professors.
Okay, so Megan has two little cute boys that are waiting for her at home. Before we end, Megan, it's close to 6:30. Oh, another question also. So two more questions.
Sure.
What advice would you give incoming students who want to get the most out of the health studies program from the very beginning?
All right, say that one more time.
What advice would you give incoming students who want to get the most out of the health studies program from the very beginning?
Okay, so the health studies program is a little different than our research track. Health studies is our one-year program that's geared towards students that are interested in going off into the medical professions. This program is set lockstep with all the coursework that our students are a part of. So our students are in courses with their cohort starting in the summer, going through till May. I tell those students that join that program, your cohort is going to be your biggest support system. Your faculty mentor is going to be your second biggest support system. Utilize them from day one. From orientation, make a couple friends, make a study group, and that's how you're going to get through the program successfully. Most of our students that have gone through the health studies program have stayed friends with some of their classmates to the point of ending up at their professional schools together. So that is my biggest thing is make sure you utilize the support system that you have within your cohort in the health studies program.
Thank you, Megan. For those interested in healthcare, is it possible to take courses like anatomy or other healthcare courses while in this program, along with the required coursework?
So this is case by case, and you would work with the program director and your graduate academic coordinator. We've had students do that in the past. We've had a student who was interested in eventually going to a MD, Ph.D. type program after they graduated. And so they doubled up a little bit, but they were able to take a couple of courses to help strengthen their academic application for medical school.
Thank you so much. Thank you, Megan. We appreciate it. We know you have the boys to go to, so we appreciate your time. And thank you again, Briana. This was, again, fascinating and amazing to hear your research, and we're proud of you. And thanks to everyone who attended today and joined us. If you would like to learn more about the Department of Biological Sciences and have more questions for Megan, please make sure that you register for our upcoming virtual open house on Monday, March 30 at noon Eastern Time. You may have already received an email about it, but if not, then the registration link will be sent in a follow-up email within the next few days. So thank you again for being with us, and we hope to see you at a future session of the Halmos College Virtual Lecture Series. Have a wonderful evening, everyone. Bye-bye.